So last time we were talking about mutations and causes of mutations. We said that mutations can and do occur spontaneously, but that a mutagen, a chemical or physical agent which increases the rate of mutation, can also be used or will occur. And so in general, the spontaneous mutation rate for prokaryotes is around 1 in every 1 billion replicated base pairs, or 1 in every 1 million replicated genes. So that's on average how often you would get a mutation. So not very often, but they do happen. And of course, mutagens can dramatically increase the rate of mutation, up to 1 in every 10,000 replicated bases, or 1 in every 1,000 replicated genes. And so there are times when scientists actually deliberately try to mutate genes in bacteria to study the effect of that mutation. So we can selectively mutate certain genes and then study the effects of those mutations. So when it comes to genetic transfer, of course, we're all familiar with vertical gene transfer from parents to offspring. And so with bacteria, it works that same way. When the bacterial cell divides, each daughter cell is genetically identical to the parent cell that produced it. And so, we're all familiar with that kind of genetic transfer. But prokaryotes are special in that they can acquire genes in the same generation from other bacterial cells. So what this means is that the bacteria don't have to take the time to mutate and change and evolve. They can essentially acquire a trait instantaneously from other members of their own species or from completely unrelated species. So we call it. And as we're going to see, there are 3 types of horizontal gene transfer. And this is wonderful for the bacteria, but it's bad for us, as we'll see. But the idea is that bacteria can acquire a gene in that same generation. They can manifest that characteristic and then pass it on to their offspring. So that's something that they can do that we can't. So the first mechanism, transformation, we'll talk about some famous experiments performed by Frederick Griffith. And so he worked with 2 types of Streptococcus pneumoniae bacteria. He worked with one type that produced a capsule. So we had encapsulated Strep pneumoniae bacteria. And the other Strep pneumoniae did not produce a capsule. And he used mice as his test subjects. So his first experiment, he injected mice with live encapsulated Strep pneumoniae bacteria, and the mice died. And of course, he found live encapsulated Strep reproducing inside the mice. But this is no surprise to us, right? I mean, what do you guys know about capsules? What can you tell me about a bacterial capsule? They are protective, right? They help to protect those bacteria from the immune system, specifically from phagocytosis. So these Strep pneumoniae were able to resist, right, the mouse's immune system and kill the mice. So we've just proven that live encapsulated Strep pneumoniae kill mice. Next experiment, we take the Strep pneumoniae that do not have capsules, inject them into the mice, and the mice survive, right? Because the immune system of the mice is able to fight off those unencapsulated bacteria or non-encapsulated bacteria, right? So live Strep without capsules do not kill. And then what Griffith did is he took the live encapsulated Strep and he heated them until he killed them. So he had dead Strep with capsules. He injects them into the mice. No surprise, the mice survive because the bacteria were dead, right? So live encapsulated Strep kills, live non-encapsulated Strep does not kill, and dead encapsulated Strep does not kill. So for the last experiment, what Griffith did is he took the live Strep without capsules and the dead strep with capsules. He mixed them together and injected that mixture into the mice, and the mice died. And what he found were live encapsulated strep reproducing in the mice. But he injected them with live non-encapsulated strep and dead encapsulated strep. And we just saw here, live strep, no capsules, do not kill. Dead strep with capsules do not kill. So both of these do not kill. Mix them together and put them in the mice, and they kill. And what they find are live encapsulated strep reproducing inside the mice. So what's going on here? How can you take two things that do not kill, mix them together, and now they kill? And what you find reproducing is not what you injected into the mice. Is it magic? So what do you think happened? Besides the one-word transformation, what do you think happened? Nope, no cross-contamination. Well, I think I know what you're going for there, but that's not how you would say it. So we have two types of strep which do not kill until you mix them together, and then they kill. And the strep that you...
sorry.
Yeah, go ahead, Rachel.
I was gonna say maybe the live non-encapsulated bacteria was able to take on the genes of having a capsule, and they were able to make themselves a capsule, and then they became.
And that's what happened. So the dead strep, which have a capsule, they were dead. They began to break down, break apart. Their chromosome broke apart. There were random pieces of DNA floating around. And the live strep, without a capsule, absorbed one of those pieces of DNA. And that piece of DNA had the gene to make a capsule. And so the non-encapsulated strep cell that absorbed that gene could now produce a capsule and survive the immune system of the mouse and reproduce and ultimately kill that mouse. So we call that process transformation. It's a naturally occurring process whereby bacteria can pick up random pieces of DNA from the environment. So they just absorb them. Once those pieces of DNA are absorbed, they may be degraded by the bacterial cell, or they may actually, code for something that's not useful, or it may only be an incomplete gene, so it serves no purpose, or maybe it's a eukaryotic gene. If it's a eukaryotic gene, well, that gene has introns, so it's not going to form a useful product. But this process is a natural process whereby bacteria can acquire new characteristics by absorbing pieces of DNA from the environment. And that's something we cannot do, right? So we don't absorb DNA from the environment, but bacteria can. And they do it with short linear pieces. But in the lab, we do not work with short linear pieces. We work with larger circular pieces of DNA called plasmids. And bacteria do not normally naturally absorb plasmids. They're too big, they're too difficult. So in the lab, we force the bacteria to absorb the plasmids. We do that by making them what's called competent. So a competent bacterial cell is capable of absorbing a plasmid. And in the lab, we do that with heat shock. So we actually heat the bacteria to 42 degrees Celsius, and they're floating in a solution of calcium chloride, and that together helps make the membrane more permeable so the plasmids can get in. And we'll actually talk about this next week in lab. We talk about the transformation experiment that we would normally do. But transformation is a natural process that occurs with short linear pieces of DNA. We can manipulate the bacteria and make them absorb plasmid DNA. But back to the natural transformation. So here we have a bacterial cell, some random pieces of DNA in the environment, and here the cell has absorbed one. Now, normally linear DNA is actually going to be attacked and degraded by the bacterial cell. because linear DNA usually represents a virus, a bacterial virus trying to infect that cell. And so part of the bacterial cell's defense, sort of part of its immune system, quote unquote, is to destroy linear DNA. So in order for this linear DNA to survive and serve any purpose, it must recombine with the host chromosome and integrate. So if that happens, then the DNA survives. It's now part of the bacterial chromosome, and if it codes for a beneficial trait, this cell now has an advantage. It can survive better. So when it reproduces and divides, every daughter cell has a copy of that gene. So this cell has just immediately acquired a new characteristic. It did not have to mutate. It did not have to evolve. Just boom, it's got that characteristic. And so bacteria can do these kinds of things. And like I said, they can absorb all different types of DNA from different organisms. Although most of the time the DNA is going to be of no benefit whatsoever, every once in a while it is beneficial. And that allows bacteria to adapt and survive. But they can simply acquire new characteristics. Humans cannot do that. Now, conjugation is another type of horizontal gene transfer. It's sort of pseudo-sexual reproduction in bacteria. As we're going to see, two cells will actually get together. They're going to hook up. And a copy of DNA is going to be transferred from one of those cells to the other one. So it is possible for bacteria to acquire genes directly from another cell. It is plasmid mediated. We're going to focus on gram-negative bacteria because they use the structure called a pilus. You may remember several weeks ago we talked about pili and fimbriae, right, protein structures that are sticky. Here we're going to see the pilus, right, singular, reach out, grab another cell, and pull it close together. We'll also mention gram-positives because they do conjugate. They do not use a pilus. We're not going to go into any detail about gram-positives. We're just going to mention that they can conjugate. But we're going to talk a lot about the gram-negatives. So the gene that codes for the pilus is called the fertility gene or the fertility factor. It's typically located on a plasmid called the F plasmid. If a bacterial cell contains the F plasmid, we designate that cell F+. So that means it's fertile. It can produce a pilus and conjugate. The cell that an F+ conjugates with is not fertile. It does not have a plasmid, so it's F-. So an F+ conjugates with an F-. So the F+ cell has that plasmid floating around with the gene for the pilus on it. But sometimes the plasmid will integrate with the bacterial chromosome. We call those cells HFR. Those cells are still fertile. They can still conjugate with an F-. But as we'll see, the end result for the F- cell is different, depending on whether it conjugates with an F+ or an HFR cell. But the purpose of the pilus is just to reach out, grab an F- cell, and pull those two cells close together. Once they touch, a conjugation bridge forms, and that allows the F+ or the HFR cell to send a copy of DNA over to the F-. So this is an older diagram because often our students still buy the older textbook, and it's not entirely accurate. Where it says sex pilus should actually be conjugation bridge, because the older editions of the textbook actually used to say that the DNA was transferred through the pilus, and that was not correct. And so they eventually did correct the textbook. But all the pilus does is pull the two cells close together. Once they touch, then the conjugation bridge forms. When an F+ conjugates with an F-, a copy of the fertility plasmid is sent over to the F- cell. Now the F- cell has the fertility plasmid, so it becomes F+. So a copy of the fertility plasmid is transferred from the F plus cell to the F minus, making the F minus F plus. So now we have more F plus cells. And the question you should be asking yourself is, well, okay, so what? So we're making all these cells fertile so they can conjugate. Who cares? Well, the plasmid has more than just a gene for Apellis on it. There are other genes on the plasmid. Those other genes could be very beneficial. And so it is a way to share those genes. But an F plus conjugating with an F minus makes the F minus become F plus. So any questions on that? So if you're using an old edition of the textbook, it's going to say something that's incorrect. And then they began to modify and just leave it ambiguous. And eventually they changed it. But the whole point of the pilus is to reach out and grab an F minus cell and pull the two cells close together. Once they touch, a conjugation bridge forms. Now, what happens if an HFR cell conjugates with an F minus? Well, in order for an HFR cell to be produced, the fertility plasmid has to integrate with the bacterial chromosome, so a recombination event occurs, and now you have an integrated plasmid. As we said, this cell, the HFR cell, is still fertile, it still makes a pilus, it still conjugates with an F minus, but what happens to the F minus is different. So when the HFR and the F minus cell conjugate, the copy of DNA that gets sent over is chromosomal. So what reaches the F minus cell is some chromosomal DNA from the HFR cell. The integrated plasmid is actually at the end, and in order to transfer a complete copy of the chromosome, including the plasmid, would require those two cells to remain hooked together for at least 90 minutes. Remember that many bacteria can divide in as little as every 20 minutes. So it's not going to work to stay hooked together with another cell, right, for that long. So the F minus cell remains F minus. It is a recombinant cell because it has acquired some new DNA, which may or may not be beneficial, but it's chromosomal DNA from the HFR cell. It does not include the fertility plasmid. So when the HFR conjugates with an F minus, the F minus cell stays F minus, but it has acquired new DNA, so it's a recombinant cell. This new DNA may contain a beneficial trait. So the whole purpose is for bacteria to share DNA, to share traits, and spread them. So it's good for the bacteria because they can acquire this trait immediately, right? And then they have it and can pass it on to their offspring. So any questions on the F plus and HFR cells?
Are you able to explain the slide we're on one more time?
Explain what again?
The slide that we're on with the HFR and the F
Yeah,
minus.
so we've got the HFR cell. Remember, the plasmid is integrated. The HFR cell still produces a pilus, reaches out to grab an F minus, pulls the F minus close together, a conjugation bridge will form, and so the copy of DNA that gets sent over from the HFR cell actually is chromosomal, right? The transfer direction goes like this, and so the first thing to be transferred over is chromosomal DNA. And because the two cells do not stay hooked together for very long, only a small piece of the chromosomal DNA is copied and sent over. And so the F minus cell stays F minus. It does not get the gene to make a pilus, right? Because that DNA would have been transferred over last. And that takes way too long, at least 90. So bacteria cannot stay hooked together that long. And so the HFR cell sends over a copy of some chromosomal DNA, and the recombinant cell It's still F minus, but it's got some new DNA, which may be beneficial. So does that clarify that?
Yeah, thank you.
So again, we're just going to mention the gram-positives can conjugate. They do not use an F plasmid. They do not use a pilus. So exactly how they do it may vary from species to species. So we're just going to mention they do it, but not go into any detail about it. Our last mechanism of horizontal gene transfer is transduction. So transduction is mediated by a bacterial virus. So we'll talk about viruses, I think, starting next week. But the idea behind a bacterial virus is that the virus lands on the bacterial cell. It actually will kind of drill a hole through the cell wall and inject its DNA, which is linear DNA. And so many bacteria will try to destroy this linear DNA, but sometimes they can't. And so the viral DNA actually takes over. The bacterial chromosome breaks apart because the virus only wants its DNA to be replicated and transcribed and translated to make new viruses. So all the parts and pieces come together, they assemble. Normally, the viral DNA gets packaged inside that new virus, but because there are random pieces of bacterial chromosome floating around, sometimes the bacterial DNA gets incorporated into the virus. And so when this virus leaves this host cell, because this host cell is going to die, and this new virus goes and infects, quote unquote, another bacterial host cell, the DNA that gets injected is bacterial DNA from the previous host. So this bacterial cell is not actually being infected with a virus, right? The virus is actually carrying bacterial DNA from the previous host, and that's what gets injected into this new host cell. And again, this DNA may or may not be beneficial. It may contain a beneficial gene or it may not. But if it is a beneficial gene, then this new host cell, lucky for it, has acquired a new characteristic which can help it to survive. But the whole process here is mediated by a virus, and the virus accidentally picks up a piece of the bacterial chromosome from the host it's infecting, and then it transfers that piece of bacterial chromosome to the next host. Any questions on transduction? So we can exploit all three of these mechanisms in the lab, although by far the easiest mechanism to use is transformation. And we'll talk about that in lab next week. And it really is ridiculously simple to put genes into bacterial cells. And so on that question about cloning a eukaryotic gene into a prokaryotic cell, so the last step is to take that clean gene, stick it into bacteria using the process of transformation. And it's actually really easy to do. And when they discovered these techniques back in the 1970s, there was great concern because it is so easy to stick genes into bacterial cells. What if you're evil and you want to put a nasty gene into a harmless bacterium? It's very easy to do. So there was great concern about bioterrorism. Fortunately, that kind of thing's never really happened. It is ridiculously easy to stick genes into bacteria. Now, in order for the gene that's picked up by the bacteria to be useful, it has to recombine with the bacterial chromosome. It has to become part of that circular chromosome. So recombination is, again, a natural process. All cells are capable of genetic recombination. I just want to review some basics with you and review some of your biology 150 material. So here we're seeing an example. Are we looking at a prokaryote or a eukaryote here? What are we talking about here? Prokaryote or eukaryote? And how do you know?
Is it a eukaryote? Eukaryote
Yep,
because it's linear?
right. So we have linear chromosomes. And as you may recall, adjacent sister chromatids can break off, switch positions, and reattach. So we all remember that. And we're seeing that here with crossing over, where the adjacent sister chromatids will break, reattach, right? Seems pretty dangerous having a sister chromatid break off. And one of its arms breaks off. What if it floats away? Right? That would be bad. And yet this happens all the time. What's the benefit? What's the benefit of crossing over? Switching arms with the adjacent sister chromatid. What's the benefit? Right, genetic variation and diversity. Right, so mixing up the genes. In particular, this happens when we're making eggs and sperm. So is this mitosis or meiosis? Meiosis. Is it meiosis 1 or meiosis 2? It's meiosis 1. Prophase 1, metaphase 1, anaphase 1, or telophase 1? Prophase 1, right. So don't forget all that stuff you learned in 150. It will come back to haunt you. So when it comes to plasmids, we can group plasmids into 3 different categories. Our conjugated plasmids, like the F plasmid, a dissimilation plasmid, which is just a plasmid that's got genes that allow a bacterial cell to use different substrates for energy, and the R factors. And R stands for resistance. And this is the thing that's really bad for us, really good for the bacteria, because the bacteria do not have to mutate and evolve resistance to antibiotics. They can simply acquire that gene from other bacterial cells. And so they can and do easily share their resistance genes. And like I said, that's great for them. It's really bad for us. But when we talk about antimicrobial drugs, we'll actually see a really cool video which shows just how quickly bacteria can mutate and change to become resistant. But resistance is a huge problem because it's so easy for bacteria to do. I will talk about that later. So plasmids, small circular pieces of DNA. You can buy many commercially produced plasmids, dozens of different plasmids from different biotech companies. And if they don't have what you're looking for, you can get one custom made. The plasmid that we're going to talk about in the lab next week is PGLO, or PGLO, a custom designed and manufactured plasmid. And we'll see what that does next week. I want to mention a few words about transposons, because transposons are an example of a bacterial intron. It's just not like the other introns that eukaryotes have, but it is considered to be an intron. But transposons are pieces of DNA that actually cut themselves out of a chromosome and move around and insert themselves somewhere else in that chromosome, or in a different chromosome if it's a eukaryote. So these are jumping genes, mobile genetic elements, pieces of DNA that cut themselves out and move around in your genome. And these were first identified by Dr. Barbara McClintock, who was working with corn. And she won a Nobel Prize for this. And since then, we've discovered lots of different transposons in many different organisms, in both prokaryotes and eukaryotes. In prokaryotes, the transposons will often jump back and forth between the bacterial chromosome and a plasmid. But in eukaryotes, it's not entirely clear where they always go. But again, here we have something that seems to be of concern. If you had a transposon insert itself into the middle of a very critical gene, well, that gene's no longer going to function. And that could be very, very bad. So then why do these transposons exist? What possible use could they be? So how can these mobile genetic elements be beneficial? It sounds pretty dangerous, but they exist, and they have for a very long time. In fact, there are probably some leftover pieces of viruses that infected us who knows how many thousands or millions of years ago. So how can they be beneficial? Come on.
Can they be used to alter DNA?
Yeah, but why would we want that? What's the magic word? Increasing, right, diversity, genetic variation. And so the question you guys often ask, so what? Who cares about all this stuff, right? Well, I just told you that bacteria can acquire a new characteristic almost instantaneously, so without having to mutate and evolve, especially when it comes to antibiotic resistance. So that's great for them. But for us, because we've learned about transformation, conjugations, transduction, we can use those mechanisms to genetically modify bacteria and use them as factories to make things for us like insulin, or we can use them to study genes. So these techniques, these three mechanisms, are actually very useful to us in terms of biotechnology. So any questions on genetics? Because we are going to start and get through classification.
Please, Professor, can you explain the implications of gene transfer among bacteria again?
Remember, they can share resistance genes, and that's great for them. It's bad for us. But we can also use these techniques to genetically modify bacteria and use them as factories to make things, or use them as little laboratories to study the effects of genes and mutations on genes. Is that good, Christiana?
Yes.
Okay, so now we're going to start classification. So we mentioned some of this in the very first lab, or the first lecture, the introductory lecture, and we'll go into some more details here. But taxonomy is the science of classifying organisms. The benefit to this is that whenever you are presenting a paper or submitting a paper or having a lecture or some kind of presentation seminar, we all use the exact same names. No matter where you're from, we all use the exact same names for the organisms. So these are universal names. And if you have an existing classification scheme, when you find something and you don't know what it is, you can try and match it to a known organism in the classification scheme. If it matches, you've identified that organism. If it doesn't match, you've discovered something brand new. And he or she who discovers something new gets to name it. So systematics or phylogeny is the study of the evolutionary history of organisms, so we can determine evolutionary relatedness by studying the phylogeny of different organisms. I just want to mention the all species inventory, which is supposed to be completed by 2025. Nowhere near close to happening, and it probably never will. The idea was that scientists wanted to try to identify and catalog all the forms of life on the Earth and do that by 2025. It's a nice idea, not going to happen, not enough money, not enough funding. And remember, most life on the Earth is probably microorganisms, so trying to find everything, wherever it might be, and then to classify it and name it is just not going to happen. It may never happen, but it was a nice idea. A little bit of history, and this is just FYI. In the beginning, when humans first started to classify living things, they only had two groups, plants and animals. So you were either a plant or an animal. When they discovered these microorganisms, bacteria and fungi, they were put into the plant kingdom, even though they truly, were not plants, but they also were not animals, so they stuck them in the plant kingdom. Later, they proposed protista for the bacteria, protozoa, algae, and fungi. And the problem with doing that is that you're mixing prokaryotes with eukaryotes, right? Because bacteria are prokaryotic. Everything else there is a eukaryote, so that really wasn't good. In 1937, the term prokaryote was really first used, or to refer to the bacteria because they had no nucleus. In 1959, they proposed the kingdom fungi and then moved the fungi into that kingdom. 1968, they proposed the kingdom prokaryote, which never really caught on. It was never really used, at least not for very long. In 1978, they discovered the archaea, that other major group of prokaryotes. So one of the problems with classification is it's constantly changing. You have to get a lot of scientists to all agree to a particular name or a particular classification group. And that's not easy to do. So these things are often proposed, never adopted. But we do have 3 domains of life. And remember, domain is the largest grouping now. It used to be kingdom, but now it's domain. And 2 of the 3 domains are prokaryotic. We have domain archaea and the domain bacteria. And then lastly, the domain eukarya, which has all the eukaryotic organisms in it, plants, animals, fungi, and protists. Now, when they first discovered the archaea, they called them archaebacteria because they were prokaryotes, and so they just grouped them in with the bacteria, but they noticed some consistent differences from the true bacteria. Archaea do not have cell walls made of peptidoglycan. So there's no peptidoglycan in the archaea. So that was a major difference. Another difference is kind of a fine difference in terms of chemistry, and we're not going to go into this in any detail other than to mention, but you do recall that the lipids are made of glycerol and fatty acids, the phospholipids in the membranes. And so the glycerol is bonded to the fatty acids with an ether linkage in the archaea. But in everything else, in the bacteria and the eukaryotes, the glycerol is bound to the fatty acids with an ester linkage. So these were 2 consistent differences. And so they decided to separate the archaea from the true bacteria and put them into their own domain of life. So here we see a kind of simple phylogenetic tree. And of course, a tree has branches, right? And so the idea is that the more closely related 2 groups of organisms are, the closer their 2 branches will be. So if I ask you just in general, looking at this tree, animals, to what other group are the animals most closely related? Just looking at this tree. What other group would you say is more closely related to the animals? Right, the fungi. And so not only are we close to that branch, but wherever you have a branch point, like we see here, that branch point represents a common ancestor. And so that ancestor diverged to create, right, the other branches. So here we share a common ancestor with the fungi. Now, of course, we're not really closely related to fungi, but at the cellular and molecular level, we are. And as I'll talk about when we do antimicrobial drugs, trying to find effective drugs against fungi was not easy because fungi are eukaryotes. We are eukaryotes. So our cells have a lot of similarity. And trying to find a drug to selectively target the fungi took some time, but we'll talk about that later on. Now, if I were to ask you, animals or eukaryotes in general, are eukaryotes more closely related to the true bacteria or the archaea?
To the archaea.
Right, because here we have a common ancestor, right, that gave rise to the archaea and to all the eukaryotes. So yes, technically, we're more closely related to archaea than we are the bacteria, but we're not closely related at all. But this is how you look at these trees. And there was some common ancestral prokaryote that gave rise to everything. So when we look at mitochondria and chloroplast, we see some remarkable similarities to bacteria. For one, mitochondria, chloroplasts, and bacteria all have a circular chromosome. And remember, eukaryotes have a lot of long linear chromosomes. And in order to keep those long linear chromosomes organized, you need histone proteins. But if you have a circular chromosome, there's no histones. When it comes to ribosomes, mitochondria and chloroplasts have the same type of ribosome as prokaryotes. Eukaryotes have a different ribosome. And when it comes to reproduction, we already learned that bacteria simply divide into binary fission. And so mitochondria and chloroplasts also divide in two through binary fission. But eukaryotes have that complex process of mitosis and cell division. So where do you think mitochondria and chloroplasts came from?
Probably from prokaryotic cells.
Yep, from bacteria. So we call that theory the endosymbiotic theory, which you should hopefully remember from 150. But the idea is that a very long time ago, one cell swallowed another cell. But the cell that got swallowed was not eaten. It was not destroyed. It actually survived inside of that host cell. And they learned to live together symbiotically. And over a very, very long period of time, many eons of time, the cell that got swallowed specialized for energy production because all of its other needs were being provided for by the host cell. So it didn't need to do all the things that a cell would normally do anymore. And so the symbiont specialized to become a mitochondrion or a chloroplast. A real life example that exists today is this organism that we see here. And this organism is a real one. Its name is Cyanophora. Oh, it's an R in there. Cyanophora paradoxa. Because it's a paradox. You have a eukaryote with a prokaryote living inside of it. And it happens to be dividing right now. But this is a real life organism that exists today. And this may represent what happened, countless eons ago, where one cell swallowed another. And that led to the formation of mitochondria and chloroplasts. So the endosymbiotic theory does try to explain the origin of mitochondria and chloroplasts. One cell swallowed another a long time ago. They learned to live together. And the cell that got swallowed specialized for energy production, either as a mitochondrion or a photosynthetic chloroplast. So we can thank that rare event for leading to the evolution of eukaryotic cells and then multicellular organisms, right? Because without mitochondria and chloroplasts, there would be no complex multicellular organisms, because a complex multicellular organism needs a lot of ATP.
So did plants also rise from that?
Yep, plants both have mitochondria and chloroplasts.
Okay.
So when it comes to names, these are the two names that are on your handout, but just some examples, because the names do tell us something. In the case of Klebsiella pneumoniae, it's named after the scientist Edwin Klebs and the disease that causes pneumonia. Pfisteria piscicida, named after Lois Pfister, and a disease that's caused in fish, because piscicida comes from Pisces. Salmonella typhimurium, we're going to see next how this has been reclassified, but originally named after the scientist and the disease that it produced in mice. Streptococcus pyogenes, so it's genes, not genes, so pyogenes. Of course, you know what streptococcus means, chains of grapes, and the pyogenes refers to pus forming. Staph aureus, again, you know clusters of grapes and the golden pigment. Legionella pneumophila was a newly discovered organism back in the 1970s, I think. And there was a conference, the American Legion Conference, and a bunch of people all got the same respiratory infection at this conference. And so whenever you have a large number of people all getting sick at the same place at the same time, that's a red flag. That tells you something is going on. And what they discovered was this brand new organism. New because they'd never known about it before, although obviously people had been infected with this organism before, but it was just bacterial pneumonia. They treated it with an antibiotic and never thought twice about it. But when all these people got sick at the same time in the same place, they took a look to find out what it was. And so there's this organism, Legionella pneumophila, which infects the lungs, and it lives in water. And so air conditioners, the condensation from air conditioners, that can create a problem. And then the organism is, blown throughout the ventilation system. Large water tanks on residential buildings and things, it's possible they can get contaminated with Legionella. And then people are taking showers or drinking from water fountains and things like that. So it was new only because we never noticed it before. But now we know. Salmonella. All the different species of Salmonella, or at least most of them, if not all, have been reclassified as Salmonella enterica. So that's the species, or the specific epithet is enterica. And all the old species names are now varieties or serovars. So Salmonella typhimurium is now Salmonella enterica variety typhimurium. So all those old species of Salmonella are now varieties or serovars, and the species is all enterica. And so this actually makes writing the name a little confusing, because you'll still see all these old names. They're still commonly used, right? Because it's hard to stop using a name that you've been using for decades. But when you write this now, this is what you write. And when I first saw Salmonella typhimurium written that way, my reaction was, what is wrong with these people? How do they not know how to write a proper genus and species? Well, typhimurium is no longer the species, it's the variety. So the way you write this now is you capitalize and italicize the genus, but the variety is capitalized, it's just not italicized. So it looks very wrong and very confusing to have this, but that's how they do things now. for this particular genus. So you will still see the old names, Salmonella, whatever, Typhimurium, Cholera suis, all different kinds. Now it's Salmonella, Enterica, and then whatever the variety is. So names change. Organisms are reclassified all the time. When I first started working here, Enterococcus faecalis was called Streptococcus faecalis, but it got reclassified. So names do change. Organisms do get reclassified, especially as we learn more genetic information and realize that they should be put into different groups. So classification is frequently changing. Now, when it came to all the different taxons, all the different groupings, remember it's domain, phylum, class, order, family, genus, species, or Dam King Philip came over from Spain. When all these names were created, all these different groupings, they're based on mostly observable characteristics. Well, with prokaryotes, there's not very many observable characteristics. So the nomenclature really is kind of artificial, which is why we're not going to bother learning it. We will only refer to bacteria by their genus and species, and sometimes we'll use the family. As for the rest of this, if you're interested, it's in the book, but we're not going to waste our time on it. So, species. What makes a species a species? Well, the traditional definition was, organisms that can interbreed and produce viable offspring. Well, bacteria don't technically interbreed, right? So they had to come up with a broader definition of species, a population of cells with similar characteristics. That's very broad, right? And within a given species, you have different strains, right? Because there are genetic differences between members of the same species, like E. coli 0157H7. It's E. coli, but it's a nasty form of E. coli. It's a dangerous form of E. coli, but it's still E. coli. So then there's a question that I've always had. So how many mutations, how many changes does a species have to get or have to have happen to it before it becomes a different species? Right? Because bacteria have a lot of similarities, right? They're genetically very, very similar. And there's just, certain differences genetically. So how many of those differences do there have to be before that organism becomes something else? And that's a question that I've never found an answer to. Right? Because we do have a lot of different strains of the same species. And we just learned that bacteria can acquire characteristics, right, by picking up genes from the environment. So how many new genes does it have to pick up before it becomes a different species? Right? So I don't have an answer for that. A viral species is similar to the bacterial species definition, a population of viruses with similar characteristics and that occupy a particular ecological niche. So that means a particular host that they infect. So just to refresh the domain eukarya, we have animals, plants, fungi, and protists. They're all eukaryotes. Animals are multicellular. We do not crunch when we move, so no cell walls. We're chemoheterotrophs. What does chemoheterotroph mean? What's a chemoheterotroph?
You use chemicals and organic compounds.
Right, so all different kinds of organic compounds for carbon and energy. Plants are multicellular. They have cellulose cell walls, and they're photoautotrophs, so light and CO2. Fungi are also chemoheterotrophs. They can be single-celled or multicellular. Their cell walls are made of chitin, and they can develop from spores or from fragments. And protista, whatever is a eukaryote that doesn't fit into the fungus, plant, or animal kingdoms is put into protista. So a large, very diverse group. And many of these organisms are being moved out of Protista and put into their own classification. So again, things are always changing, but Protista is where you would stick any eukaryote that doesn't fit anywhere else. So it was a very, very diverse kingdom. But they've been moving things out and reclassifying them. So for the prokaryotes, I would love to be able to spend time talking about archaea, but we just don't have that time. They're very interesting microorganisms. They're often referred to as extremophiles because they love or need extreme environments like extremely high salt, extremely high temperatures. Methanogens are not really very extreme, but they do make methane gas. So. We mentioned Berge because he was the first scientist to assemble all the known information about all the known microorganisms and put them into very large books. And so the Berge's manuals were the gold standards. And we actually have an antique copy in lab that we kept. Of course, today there's lots of different sources for identifying microorganisms, and it's all digital and on computers, but Berge was the first one to do it. All of his data were based on observable characteristics like microscopy, staining, biochemical testing. These days, there's all kinds of genetic information available. Now, when it comes to identifying, and we'll focus more on this after midterm in lab, but of course, we stain the organism to see its shape, arrangement, and various other characteristics. We perform a variety of biochemical tests. And remember, the purpose of a biochemical test is to see if an enzyme is present. And the way we know that enzyme is present is by finding the end product of that enzymatic reaction. A way to do a lot of biochemical tests all at once is to buy these plastic tubes. These plastic tubes have many chambers full of different kinds of agar. You would get a sample of bacteria on the end of the needle and pull the needle and push it back and forth to inoculate all the different chambers, incubate it, and the next day or 2 days later, you get color changes, and you score those results and obtain a numeric code, which you then look up. And that's how you could very quickly and easily identify an unknown microorganism. We actually use these kinds of things for several semesters, but they didn't work as well as we'd hoped. There were problems with using them, and they were also very expensive, so we stopped using them. And it went back to the old-fashioned way of doing, tube by tube and plate by plate tests. You guys are spared all of that this semester because we're not having labs, but we'll talk about them. If you want to get more sophisticated, you can perform serological tests. So serology means antibodies. So we're using antibodies and antigens, so usually proteins. If the antibodies stick, we can see that in different ways. With slide agglutination, agglutinate means to clump together. So you take your unknown sample, you have an antibody to a particular pathogen, you mix it with the sample. If the sample clumps together like we see here, that's a positive test result. You've identified that pathogen. If there's no clumping, then it's something else. A common slide agglutination test is blood typing. So you have antigens on your blood cells, either the A antigen or the B antigen, and we have antibodies to A and B. And so you mix those antibodies with your blood. If it clumps, you're positive for that particular antigen. If there's no clumping at all for anything, you're type O, right? Because you don't have those antigens. Now, ELISA is a very important test. And here we see an example. And what ELISA stands for is enzyme linked immunosorbent assay. So enzyme-linked immunosorbent assay. So you have these plates with these plastic wells, and you have antigen in the well. So whatever kind of antigen that you're testing for is placed in the well. And you can get these commercially produced to test for different things like HIV or COVID-19. And so the idea is you take some serum from a patient, you add it to the well. If the patient has antibodies to the antigen, they will stick to the well. So you add a second antibody which sticks to other antibodies. And the second antibody is labeled, usually with a chemical that will produce a color change. And we see an example here where the color change is purple. And so the idea is, if the patient has antibodies to a particular antigen, those antibodies stick to the wells. Then you add a second antibody which sticks to the first antibody. If there's no antibody stuck in the wells, then no chemical reaction will occur. And the darker the chemical reaction, the more strongly positive the test result is. So the more antibodies are present. So we got a lot of strong positives here. And we have some weaker ones, right? The weak ones might have to retest because you can't be sure. But ELISA is a very powerful tool. It's very commonly used. You can make your own plates, or you can buy them commercially prepared, to look for different kinds of antigens, or to look for different kinds of antibodies to antigens. So that's ELISA. Any questions on ELISA, or ELISA, as some people say? Western blot involves electrophoresis. So hopefully you guys remember agarose gel electrophoresis. We normally use that to separate pieces of DNA, but we can also use gels to separate proteins. So you have a mixture of proteins and you separate them on a gel. And then you add your antibodies, which again are labeled. It may be a chemical label, it may be a radioactive label, but the antibody will only stick to the gel if that protein is present. So you can identify if protein is there, right, using this technique. But serology means using antibodies against antigens. And the antigens are typically proteins. If you want to get more sophisticated, you can do DNA fingerprinting. And this is where you do take DNA, you cut it up with restriction enzymes, and then you run a gel, and you get a fragment pattern. Those patterns are fingerprints, and they're specific. So you could use this to identify an unknown organism, or you could use it to identify an unknown suspect in a crime, right? Or you could use it to identify who daddy is, right? So there's lots of things that you can do with DNA printing because the fingerprints are unique and they're specific to a given organism. Now, in order to do a DNA fingerprint, you need kind of a lot of DNA to do this. Maybe you don't have very much DNA to work with. So there is a way to increase the amount of that DNA. And that technique is called PCR, or the polymerase chain reaction. Has anybody heard of PCR before? Yep, it's a very, very powerful tool. And in fact, it won a Nobel Prize. So you have 4 ingredients in PCR. You have your template DNA, which you're going to amplify. Of course, you need the enzyme that makes DNA, which is DNA polymerase. You need all the nucleotides, right, the ATGCs, which are called DNTPs for short. I know it's not really very short, but that's what they call them. And then we need primers. So these are the only 4 ingredients you need to make DNA in a test tube. Just those 4 things. But because that's all we have, when you take your template DNA, it's double-stranded. We need to separate the strands. We have no helicase, right? Because we only have one enzyme, DNA polymerase. So we're actually going to use heat to separate the 2 strands. Now we have 2 templates. Our primers will bind, and they create a region of double-strandedness, which allows DNA polymerase to hop on and start to replicate. So now we've doubled the amount of DNA, right, that we started with. We heat this up. Now we have 4 template strands. And again, primers will bind, and then a polymerase will replicate, and we double the amount again. We do the same thing again. We do this for 30 to 50 cycles. Each cycle doubles the amount of DNA. So we can very quickly increase the amount of DNA that we have in really just a couple of hours, with the machines we have today. So a remarkable technique which won a Nobel Prize. But there is a problem. The temperature that we use to heat up, 95 degrees Celsius. What happens to enzymes at 95 degrees Celsius?
They can get denatured.
Right, they denature. So that would mean if you're running this process 30 to 50 times, you would have to add DNA polymerase 30 to 50 times. That is ridiculously tedious and expensive. So that's not going to win a Nobel Prize. So how do you deal with the DNA polymerase denaturing problem and win a Nobel Prize? So can you think of a way to avoid to add DNA polymerase 30, 40, 50 times and win a Nobel Prize? We actually just mentioned it a little while ago, and we'll actually talk a little bit about it in today's lab. But any idea how to deal with the denaturation problem? No ideas? Ruby, you have an idea? Maria? Craig? Bryant? Clarice? Any ideas? Nobody wants to make a lot of money? So we have to heat the DNA up to 95 degrees Celsius, right? That's only 5 degrees below boiling. And we know that enzymes have a problem with heat, right? They denature. So how can you sort of get around that problem? Can you think of any kind of organisms where heat is not a problem? Thermophiles. And so what was actually done, a thermophile from a hot spring in Yellowstone National Park was isolated. And so because it's a thermophilic organism, it loves heat, its enzymes love heat. And so 95 degrees is not a problem for it. You add the enzyme once, it's good cycle after cycle after cycle because it does not denature. So that discovery made the process of PCR very efficient and economical and won a Nobel Prize. Of course, the scientist who did that was not a businessman. He did not really understand the potential profits involved. So he sold the patents for next to nothing when you consider that PCR is $1 billion industry. So, he was smart and not very smart. And the organism was Thermus aquaticus. And the brand name polymerase is Taq polymerase. So a thermophilic DNA polymerase was used because it does not denature with heat. You could add it once and it's good. Cycle after cycle after cycle. So everybody got that? We'll talk about PCR again in lab later on. So a dichotomous key to finish up is an identification scheme. So it's a series of yes or no choices. So it's dichotomous. You got two choices, yes, no, positive, negative. And so a flowchart is a common example and one that we'll use in lab as a way to identify unknown bacteria. You basically follow your flowchart step by step with each test result, positive or negative, and hopefully that leads you to identify an unknown organism. But we'll talk more about this kind of thing after midterm in lab. So any questions? Okay, then we'll need to stop the lecture for now. Let's start maybe 1:25, right? So we'll be back in about 6 minutes or so, and then we'll do the lab today. All right?
Thank you.
I'll see you then.